Actually, I am the co-lead of the Wildgen Habitat and Conservation Working Group. We are working with Juliet and Gilda, and currently I'm a PhD student at the ZMT in German. And with this talk, I will give a small presentation on artisanal fishing for octopus in Tanzania, checking on the currently status and management recommendations on behalf of my colleagues, Matthew, Nariman, and Deborah. So the octopus forms an important fishery for the coastal communities in Tanzania and the whole of the Wyo region. In Tanzania, the fishery have expanded over the last decades due to increasing international market demand. And overall, at least 2/3 of the catch is supplied to the international markets, and the least is supplied to the local market. In Zanzibar, the situation is quite different, of which 90% of the market is the tourists. And with this COVID-19 situation, it has brought some negative effects on the fisheries. So the catch is normally exported mainly to Europe, especially Portugal, Italy, Spain, and France, and some countries in Asia. And from Tanzania, between 2008 and 2012, about 1,500 tons of octopi were exported in different countries. And this generated a revenue of about 6.8 million US dollars, which is almost half of the total of the whole of the Wyo region. So the catch composed mainly of Octopus cyaneus. It is the largest species, but also we have Octopus vulgaris. Yeah, and the maximum weight of Octopus cyanea can grow up to 11.7 according to the reports. And the fishers use traditional fishing methods like gleaning. They use traps, trawlers, and pots. And recently, like I said, the artisanal fishers have increased their fishing intensity due to demand from the international markets, according to the report of Rockcliffe and Harris from Blue Ventures in 2016. So some of the important companies that buy the octopus from the artisanal fishers include the Bahari, Fruit de la Mer, and Tan Pesca. And between 2008 and 2012, Tanzania witnessed an increase in octopus export by 8% and valuable 1.6. And due to increasing octopus export and fishing intensities, there's already some authors like Pandu and Rockcliffe. They've raised, they posed a question on sustainability of the fishery. For instance, nearly 20 years ago, Gad and Mgaya, they reported overfishing in some sites in Tanga and in Mtwara. So a little bit on management on cephalopods is that Perry proposed three kinds of management interventions, which includes catch regulation, sex size limits, and control of fishing. And in Tanzania, like in some places, they practice a size limit of 500 grams, but it's difficult to control because you have so many artisanal fishers, and the fishing methods, they are non-selective. So for instance, a fisherman would spot an octopus in a den and then would use a spear to injure without knowing the size beforehand. So at the end, they don't find any logic to discard the octopus below recommended size. So the closure has remained as the very important alternative. It was initiated in different, but we call them experimental closure at Kilas Bosongo. The last 2 years, they were able to harvest over 17 tons of octopus. But although they increased catch, but still the benefits looks like they disappear in few days after opening. And sometimes you have fishermen coming from nearby villages, which becomes difficult to control. But of course, they can result in a strong management actions like MPA. So our study was conducted along the coastline of Tanzania, of which in one of the island called Gibondo and Beju, which is part of the Mafia archipelago. And these islands are endowed with coral reefs and intertidal ecosystems that support marine lives. Of course, as many parts of the Wio region, it's affected by monsoons, and so therefore the length and weight data were corrected during two periods between 2014 to 2015 and 2017 to 2018. They were obtained from the artisanal fishers during spring low tides. So that means that in every month that for data collections only took place 16 days in every month. So we also retrieved the fisheries and octopus trade statistics. First, we picked up the octopus catch data from the Food and Agriculture Organization. And also we picked up the export trade data from the United Nations Comtrade database. And then we use Microsoft Excel and ARA programming software. So this is how the overview of the capture data as represented by FAO and the Comtrade trade data. So we can see that over the years, the Tanzania octopus catch increased from 480 in 1990 to 5,600 in the year 2019. We see that there's a peak in 2003, and then you have a steadily increase in catch, and then steady and then sharp peaked to 5,600. So, but Tanzania, which, as you can see in the whole region, remained as important hot spots, followed by Kenya and Mauritius. In terms of trade, between 2016 and 2018, Tanzania exported over 3.4 tons of octopus with a value of 7.7 million. And we see that this is 2017, this is 2016, 2017, 2018. You see there's an overall increase in the partner countries, the markets, and the export amount. For instance, between 2016 and 2017, the amount grew by 20%, and then the export amount grew by, doubled at 2018. So this can have implications for fisheries, especially when you don't know a lot about the stock. Slightly about the length-weight relationships, we saw that Boiju had large-sized octopus compared to Gibondo, and even the same applies to the dorsal mantle lengths, were quite high at Boiju. And we can see that the value of B was around 2.16 at Boiju, and at Gibondo was about 2.28. So we've seen an increase and a peak at 2018, at 2019 of 5,600, which suggests an increased fishing effort by the artisanal fishers. But of course, we need to treat this data in cautious, because the data from FAO represent the so-called conservative underestimates. So it's probably higher than this. And the main buyers were Portugal, Italy, France, and Italy. But from this study, we see additional players and buyers include Turkey, Greece, Israel, and the Netherlands. And the previous reports by Blue Ventures, and they didn't capture that. The average weight at Gibondo was even lower than the average weight reported by the same study by Gard and Guy. So to conclude is that artisanal octopus fishery is important for livelihood. We've seen also an economy. The catch increased in Tanzania from 480 to 1,200 in 2012, and then it peaked to 5,000 in 2019. We've seen a benefits of 3.4 tons, equivalent to 7.7 millions of money to Tanzania, which between 2016 and 2018. And in the same year, we've seen an increase of partner countries up to 20 from 8 to 10. And the main buyers still remain Portugal, Italy, France, and Spain. And the length and weight data indicated that Beju was not in good state. Gibondo was overfished and required concrete management actions like closure. So we suggest a continuous monitoring of the cache data, supporting the teams on the ground, and good recording of the trading data, so that and then more research on the stocks, so that the stocks remain sustainable. These are my contacts and email in case you want to have more chats and questions. My colleagues, some of the reference, and thank you so much.
Thank you so much, Kritas, for that wonderful presentation on artisanal octopus fishery. So at this juncture, I'd like to give 5 minutes to participants. In case you have any question, please just raise your hand. And yeah, the floor is open for questions. Any questions? Yes, I see a hand from Warwick. Please unmute and ask your question.
Great. Thank you so much for an excellent talk. Really enjoyed it. I wonder if you could just fill in a little bit more on what technical measures you're thinking of introducing in terms of management. It'd just be interesting to know in terms of the closed seasons. They work well, I know, in places like Madagascar. So what is Tanzania thinking in terms of the way forward for managing the stocks?
Yeah, I mean, thank you for a very interesting question. It's a difficult one. What I can say is most of our fisheries, they lack a good data, I would say. So the very first step is to have more data, keep tracking of the stocks, which is quite, I would say, expensive. And most of the efforts for catch monitoring, like what we did in our study, it was funded, and the fundings, they are not consistent, and sometimes they dry up. So so far, to the best of my knowledge, I don't know if there is a quite very well dedicated plan only for octopus. It's like the fishery management manage fishery like overall and not specific to species. And yeah, but of course, with the raising awareness with the government, and there's quite a number of fisheries improvement projects for octopus, so that they reach the stewardship. But still, there's a lot of work need to be done. Even the closures is not very effective, because you have fishermen coming from nearby villages, and then you have the native one from the same village. They're saying why we are doing the closure anyhow. So it's a difficult one, and there's a long way to go, I would say.
Oh, thanks so much. Yeah, not an easy exercise to make it all happen.
Yeah. Thank you for that. Any more questions? I think I can contribute to a question. Due to the increase in the tonnage of octopus caught in the water, can you say that that particular fishery is sustainable to the villagers, to the
yeah, I mean, to respond to that is that most of the research that were done, it's difficult to conclude for the whole of Tanzania, but most of the research, they are taking place in very few sites, and you'll find that out of 5 sites, you have 1 site or 2 sites that are overfished. And I think it will be very important to do research in different fishing grounds of octopus, so that we have overall conclusion there of the whole of the Tanzania. But of course, different sites in Tanzania, they are in different successful stages of exploitations. For instance, the Bweju site, which had a very good weight, I would say this site, it has very few people. They live there, like around 25 fishermen, and Jibondo is almost nearby the island. So most of people can travel here, but here is much more isolated and you have very few people and less fishing effort. So you have different sites with different succession in terms of levels of exploitation. That's why we were suggesting that this catch monitoring should be done in detail and should have dedicated plan that is permanent, so that yeah, all sites, they are well monitored and we are quite in track that the fishery is sustainable. Otherwise, we will just have the data from the Comtrade and the FAO, which sometimes represent underestimates, and then we would run into problems in the future. Yeah.
Okay, thank you for that, Christos. We'll take one last question because of time from Paul Tudor.
Okay.
Yeah.
Thank you so much, Kratos. Mine is actually just a comment in line with what you just said. One will be just a recommendation, because just doing a length-weight relationship in itself does not give us the status of the exploitation rate of the octopus fishery. So one of the things would be possibly to conduct a stock assessment for octopus. But of course, we know that without specific data, using the normal data limited approaches does not apply to octopus, because of octopus mostly recruits throughout the year. So it will be difficult to conduct a normal stock assessment with a length frequency analysis. So one recommendation is to look at the catch and effort and maybe do estimate biomass. And that will give a good indication of whether the current biomass is sustainable or not. So I think it's rather just a matter of trying to look at the data and trying to see what components of the data will help you to estimate the biomass. And then that can give you a good picture of the current status of the octopus fishery. And then maybe the last recommendation would be also to ask maybe just a question. I mean, you've shown that the export for octopus fishery, octopus products is increasing. What about the local consumption? Because if the local consumption of octopus is also increasing, that can also give you a proxy of the level of exploitation that is just going on. So it's not just for the export, but it's also for local consumption. And I think adding that onto the monitoring of the fishery can give you a better proxy of what's happening to the fishery. Thank you.
Okay, thank you so much.
Thank you. I think he will look into that. So I'd like us to go to the next presenter, Erica Tovella from Mozambique. Welcome. Presentation in 10 minutes and then 5 minutes for question.
I'm sharing my screen now.
Okay.
Can you all see?
Yes. Maybe put it in presentation mode.
Okay, sorry. Is it okay now?
Not yet. Not yet in presentation mode.
What about now?
But to save on time, if you can still
Okay.
I don't know if everyone is able to see as you go on.
Okay. Morning, everyone. I'm Erica Tovella from the University Eduardo Mondlane in Mozambique, PhD student at Eduardo Mondlane, doing fish taxonomy and genetics for this complex gene, but the Letrinus. This is part of the project. It's an ongoing project. So now I'm presenting the first results that we got from the work. This is the structure of my presentation. Going to the introductions, like this is the most is considered one of the most difficult genus to study because of the conservative and they are constant, conservative in morphological and anatomical characteristics. We have this genus distributed along the Mozambique coast. Is part of one of the big coral reefs biodiversity, where we have at least 19 tree families and 794 species that are already described in Mozambique. This family contains one subfamily, 5 genera, that comprise 45 species, and this specific genus, the Trinus, contains the largest number inside the entire family. For Mozambique, it's estimated that we have 14 species that are described that in the fish guide that we are using currently. However, we have not so recent papers, but a study that was doing in the north of Mozambique at Vamizi in Cabo Delgado that shows that that they found 16 species only for that specific regions, and we assume that probably we have 17 or more species in the entire country. The main objective of this study was to do the identification based on DNA to provide an accurate fish differentiation within the genus, and to start a reference database for molecular studies in the genus. One of the premises of that DNA barcoding is that a short standardized sequence can distinguish individuals of a species because genetic variations between species exceed that within. We can, for example, through taxonomy, it's one of the most old tools used to identify species. However, if we are facing some difficulties nowadays.
Excuse me, your slides are not moving.
Your slides are stuck.
Okay. What about now?
Yeah, they're still not moving. That's why it would have been advisable if it was in presentation mode.
Do you have more than one screen, Erica? This might be the issue.
Okay. Let me try to use another screen. Can you see now?
No, still the same slide.
Okay, which one?
The first one, the title slide.
Let me stop and do it again. Is it possible to see something now?
Can you change it to presentation mode? It should be an icon at the bottom of your screen. It should be bottom right.
I did it. I'm in the presentation mode now.
On our side, it's still in the PowerPoint.
Okay.
Is there any IT person near you there? Because it's normally a challenge sometimes.
Maybe we can share it for you if that helps. Okay. Or
please. You can see this one.
Yes.
Yes,
okay.
You can see it.
What about now?
You can still see it in PowerPoint form.
What about?
Erica, we can share it for you.
Okay.
We have a copy of your presentation. So Winnie, are you able to do that? Otherwise I can do it directly.
Okay. Please.
Winnie, will you do that?
Okay, let me stop sharing this one. Okay.
Shannon, I only have Esther's. I don't have Erica's. So you can share Erica's if you have them.
Okay.
There we go. Can you see?
Yes.
Great. Okay.
Yes.
Okay. Should I start or move on?
Just let me know. You can carry on. Let me know which side you're on.
We can go. Go, please. Yes, you can continue going. Okay, I was here. No, back, please. Okay, I was saying that one of the most efficient region to identify a species is the CO1, because this region has a good power on most of animal groups for discriminations. And this species can be identified from the tiny amount of tissue from juvenile or any material for morphological identification if we face any problem. Next one. Okay, these groups are divided in three, this genus are divided into three groups, where we have species that are considered low-bodied with conical lateral teeth. The other one is high-bodied species with molariform, that is called by Carpenter, that intermediate group, and we have the high-bodied with conical teeth. So basically, one of the most important feature that differentiate this group are the teeth and the type of body. Please move on. So these are the groups that we have species for all different bodies and dental types. Yes, next. One of the problems that we are facing in Mozambique, that it's really difficult to identify these species. As I said, we are in our fish guide, we have 14. However, recent studies shows us that we probably have more, but one of the struggles we are facing is that it's difficult to discriminate one species from another one. And because we have this long coast where every place they give like a local name, and sometimes we don't have like a very precise technique to identify this group. We are facing these problems, and that's why we are pretending to use DNA barcoding to just help us to support our morphological identification. Another problem that we face was like it's difficult to identify more than 2 species using only a set of primers, where we have to follow another methodology that say that, okay, the best way to identify more than 1 species in these groups is to use a cocktail of primer where you can at least more than 4 species identification. Next. So probably this is the list that we have here. We have, for example, the first one, we still have in our guideline, where is red, we still identify the species as sanguineous, but we know that sanguineous is the same as, is the synonym of Letrinus macena. We have, for example, Letrinus elongatus, that is the same, is the synonym of Letrinus microdon. We have only these red ones, all the names are not valid anymore. So probably we are saying that we have, for example, the red ones and the black ones, while we are identifying the same species. So probably this one can lead us to having more or less species identified through this group. Another one is that we have these three, the last 3 ones. The Borbonicus is not described for Mozambique. However, in our analysis, we found that we have these ones. The miniatus, we didn't found any of them, but we have one species from our natural history collection saying that we have miniatus here. And the Letranus sp, we didn't know that we could have this one, so that's why we include here in our problems. That is something that we probably have here in Mozambique. Next one. Another issue is the difference between the climate. We are in a very big coast where we have two difference in terms of climate influence. So we have in the north, we have influence of monsoons, while in the south we have influence of cold fronts. So because of these specific areas where we are doing our studies, are some, for example, in Nyaka, Bazarutu, and Nyaka Bazarutu are from the south under influence of cold fronts, and Ibu, Mekuf, and Memba are in the north under influence of monsoons. Probably we can have two different things that we are in somehow identifying them as the same while we are dealing with different species. Next one, please. So what we did was collecting a fin of the fish, doing the extraction, doing the PCR, and afterwards doing the sequencing to have all the species barcoded and identified through the molecular analysis. Next one. Our objective was to study the variations of these species from the taking into account that we have these two big regions like the south and the north where we can find this genus. And we want to try to compare the species from one side and the species from the other side. It's like I'm saying that we want to compare north species and south species through barcode. Yes, next one. This is our study area. As I was saying, this is the north area where we have Ibu, Mekuf, and Memba is under influence of monsoons, and in the south we have Bazarutu and Nyaka that are under influence of cold fronts. Next. This is a very complicated tree because we had to include a lot of sequence from gene bank, but I don't know if it's quite visible, but we have like different colors where we have different species based on location. What we are analyzing now in our preliminary results is what we have regarding to the distributions and what we have regarding to the species. It's quite difficult to understand what we have, but next slide, please. So we have these ones that, oh, sorry, it's not possible to see the labels here, but each of these colors represent a species. So, and these are the places where we did the sampling. If you try to see, for example, here, where we are in the Nyasoru, this is one Letranus sp. It's one of the unknown species from Mozambique. And we have these ones that is the Letranus borbonicus that we didn't even know that we have here, but probably we have in a lot of different places. So the results are conducting us for more than what we expect. It's like we didn't expect to have more than one place for this, for example, the Letranus borbonicus. But it seems that we have, and we have to go again, if possible, try to collect and try to do the morphology, because we have a lot of species identified probably has Letrinus masena, while Letrinus bourbonica, for instance. And this is the species distribution, where we could see that we have more species in Ibu compared to the other parts. Next slide, please. So this is the example of, for example, these two species that depending on who is collecting and who is identifying, sometimes they can be identified as different ones. And we have these two. This one is from Iñaka, the blue one, Masena, and this the left side is the Trinus Masena from Ibu. So if someone is trying to identify them using only morphology, could be difficult. This is the small example that I'm bringing here, because we have another ones that are more difficult to identify if we try to compare to the other species. Next one. So because this is an ongoing study, we are one thing that we want to do is to perform morphology. Now we want to try to see based on morphology using a software for the shape, where we can try to see if, for example, one species that we found in the north, for instance, is the same one that we found in Iñaka. Like we have two different sites, but we have the same species that we are considering that is the same, while probably we have some small difference on morphology. We want to compare this data with the genetics ones, and we want to try to see in detail this Letrinus bourbonica species that we found more in Iñaka, but it seems that we have more in the north. Unfortunately, we cannot go more to the north because of the problems that we have now to collect more species, to try to have more things to analyze regarding to bourbonicas. We are creating a key identification for this genus through Delta, where we can give new keys for the fisheries sector to identify these species, not only the only way they are using now. And we are trying to create a database for this genus, because we don't have a lot of molecular data in Mozambique. So that's why this will be another big challenge for us to try to set up this database. Next. And thank you. Sorry for the presentation that didn't come out first of all. Thanks.
Thank you very much, Erica, for that interesting presentation on genetics. I'm sorry, because of the time, we shall not take the questions, but if you have any questions to Erica, please feel free to put them in the chat box below. So immediately we'd go to our third presenter, Esther Magondu. Kindly, if you're there, just, you can start your presentation, kindly. Esther.
Okay. Hello, everyone. Good morning.
Hello, Esther. Good morning. Kindly, let's keep time, 10 minutes.
Let me share the screen. I hope I'll manage from my side. Okay. You can see it?
Yes, we can see it. Maybe just have it in presentation mode.
Okay. It's now in presentation mode?
Yes, yes.
Okay, thank you. Welcome everyone to this presentation. My name is Esther Wairimo, and it's a short presentation of some progress of some work that I've been doing. So I work with Kenya Marine Fisheries Research Institute, Mombasa, and I'm also a student at Bonn University. I'm undertaking my studies, my PhD studies. So I'll be presenting to you a study on aquaculture this round. We are doing the title is towards recruitment of sea cucumber and cocos in an integrated culture with shrimps in earthen ponds in Kibokoni around Cliffy Creek in Kenya. So the outline of my presentation will be that I'll start with the introduction, then research objectives, the design, the results that we have achieved, the discussions, the challenges, and finally the acknowledgement. So first, I'll start with a brief introduction, or rather the background of this study. We realized that in aquaculture, the growth is going on an upward trend, according to those recent authors. And the driving force has been the demand of seafood products that is already global. So the effects to these are threats to the ecosystem due to the precautionary use of inputs that we are putting into the culture system, that is fish feeds, we have fertilizers, sometimes antibiotics when we are rearing these organisms. And the possible remedy to this situation is the concept of integrated multi-trophic aquaculture, which I'll focus on in this presentation, which has environmental, economic, and also social benefits. So towards my right side, there is a small figure there. It's actually the concept for maybe some few of us who doesn't understand the IMTA concept. The figure tries to explain about it as part of this introduction. So in an IMTA concept, we have a fed component. Like in this picture, you can see we have fin fish that are fed feed. So that is the fed component of the system. Then we have an extractive component of the system. We have, like in this case, we have seaweeds, which take up the phosphorus and nitrogen from the system. We have deposit feeders, that is the sea cucumber, which clean the system, or they take up the organic matter from the system. And we have suspension feeders like the cocos or the shellfishes, which take up the suspension, the suspended particulate matter from the system. And with that cleaning, what you expect to derive more benefits, or more benefits from the system when you catch a fish in such a system. So the statement of this problem of this, the statement problem of this study is because of raw catcher fisheries, which has led to overexploitation. So that is an existing problem. There's population increase, hence there's rising per capita fish consumption. And then there's, because of questionable aquaculture management practices, which have compromised production, then that's why we looked at doing this particular work. So to my objectives, the overall objectives of this study was to introduce an IMTA system, which I've referred to integrated multi-trophic aquaculture system for the culture of marine organisms in order to enhance overall production, profitability in small mariculture enterprises here in Kenya. So, but in this study, we focused mainly on the marine environment or marine aquaculture. So the specific objectives were to determine the viability of integrating an IMTA of sea cucumber. Those are the organisms that are used in this study, cocos and also marine shrimps. And the particular shrimps are used is the Pineas indicas. We also wanted to assess the potential profitability of the IMTA system in comparison with monoculture system in earthen ponds. And we also compared the differences in physical-chemical parameters of an IMTA system versus a monoculture system. So this map shows where the study or the study area of where this work was done. So these are where the red arrow is pointing, those are intertidal ponds along the Cliffy Creek, where there's a mariculture group that is doing some farming of different organisms. So we did set our experiment there. So, and then we had a design where we had 2 treatments with 3 replicates and we used a total number of 6 ponds where we did the replication. The first 3 we used the IMTA system, and the other 3 we used the monoculture system. So in the first treatment we had the Pineas indicas, as I've said, and then the other treatment we had the combination of Pineas indicas, the sea cucumber, which is the particular species was Horodurius cabra, and then the cocos, and the particular species was Anadara antiquata. So, and then we did, of course, the data analysis, which we used different tests as I've indicated there. So going forward is just some photos to show the experimental organisms, maybe for somebody who might not be aware of how they look like. So we have the Pineas indicas, as you can see there. This is actually where we did this study. The farmers there have just been producing and have been doing monoculture of this study using this particular species. So then we have what we introduced, the cocos, and then the sea cucumbers. So going to the results, we did, after the study, we collected data. After the capture days, as I've indicated, we did capture the organisms for 135 days. So you can see from the first graph, the weight versus the capture days. As you can see, the growth we got from Horodurus cabra, it did very well, and then which was followed by Anadara antiquata, and finally the Pineas indicas. Versus now the monoculture, where we still the same experimental period, and also the growth, as you can see, or the weight achieved, as you can see from that graph. Then the yield, I have a table showing the growth and yield parameters that were studied or that were collected from when we were we were carrying out the experiments. Both treatments. So treatment 1 was the monoculture of the Pineas indicas, as you can see from that table. And then treatment 2, it was the IMTA, which shows the different parameters, the stocking densities, the stocking weight and the harvest weight, the average fed component, average apparent feed conversion ratios, which I calculated only for Pineas indicas, because those were only the fed component of the system. That's why we are missing the two from the H. scabra and the Anadara antiquata. Then we had the net weight, individual net weight gain, the daily growth rate, and also the specific growth rate, and then the survival. And here I would like to mention, you can see the survival for H star for the sea cucumber was quite low as compared to the other organisms. It's because of we experienced some predation from mud crabs that were predating on the sea cucumber, so that's why the survival was quite low, as you can see. And also finally we calculate treatment. So on the far right, you can see the different organisms at harvest. So those were the sizes at harvest, as also shown in the table. So also during the experimental period, we carried out the physical chemical parameters of the capture water in both treatments. Where we did analysis we corrected the in situ parameters, as you can see, the DO, the temperature, the transparency, salinity, the pH, and also the nutrients. And this we compared for both treatments, and also for the different time periods, for the different times that we did the collection of the samples. And this we had to use a repeated measures ANOVA, so that we can get to know the difference at different times, what was happening in the different treatments at different times. So when we went ahead and did an economic comparison, so that at least we could get a picture of how the performance was from the different treatments, and also a way of advising the farmers from this kind of small experiment we did. So for the first monoculture treatment, you can see we got a cost-benefit ratio. The net income was negative, so there was the performance was not very good, and also the cost-benefit ratio was 0.27 in US dollars, and the total income also the net income in US dollars also for treatment 2 was quite high, because it was 516 USD, and then they performed so well with the cost benefit ratio of 1.77. So that that's how the two treatments performed. So we could say from here we derived that the IMTA treatment performed better, of course, because it had quite a number of combinations. Then on discussing this briefly, so we discussed that a comparative performance analysis between the two treatments portrayed the feasibility of farming shrimps and the sandfishes together in raw-fed brackish ecosystems. The IMTA model, which performed better than the monoculture, and this concurred with some recent studies from these two others, as shown there. Then shrimps recorded similar results for growth in both treatments, so there was not much difference. Then sea cucumbers in the IMTA treatment grew bigger and faster, with a specific growth rate of 4.1% and a net weight gain of 175 grams in the 135 days of capture period that we did this experiment. Then the policy action, now I was trying to relate this to the theme of this conference. So because of in Kenya, there have been over-exploitation of sea cucumber fishery, which has created alarm and of course a worldwide management concern, which the main reasons have been the species is high value, there's serial local depletion, and fishermen are migrating also to new areas to try and get these species. So policies have been developed that has led to these species being listed. It's actually listed as an endangered species in the IUCN, and also other revisions have listed other species like the Horodurian obelis and also the Horodurian foscogilva. And also we indicate here that management alone cannot sufficiently overcome the issue of declining population, hence the need to initiate sea cucumber farming. So besides having this organism in our IMTA setup, we were trying, because in Kenya we have not yet started capturing sea cucumber, so it was a way of trying to see if sea cucumber can be really grown in a closed environment, or how it can perform in pond environment. And actually from these results we are able now to go on with this kind of research. So conclusions, the conclusion of this study is that the findings exhibit that IMTA involving marine shrimps as fed species with cockles and sea cucumber as extractive organisms, they performed better than monoculture in terms of production, as you saw, and also economic returns were better, and also the water quality. So the results indicate the potential viability of IMTA as a raw-intensive culture system that can be adopted by coastal farmers in the region. Because the only challenge was getting the seed, because we had to rely on wild seed. But once the seed is put in the pond, like for the sea cucumber, you don't need any feeding, because they feed on the organic substances in the bottom of the pond. So actually for the farmers, that is a plus, because they'll not incur so much cost, as well as the cockles, they'll filter feed from the system. The only issue of the input that you put in the system is only the feeding for the fed organism. In this case, it was the Pineas indicas or the shrimps. So it's something that can be adopted easily by farmers. So we recommend further research on longer capture periods and with different species combinations with commercial value, which need to be explored to assess suitability for capture and application in IMTA systems. So, and also need for government support in terms of capital and policies directed towards domestication and capture of the species for sustainability, I think.
Thank you very much, Esther, for that wonderful presentation on the IMTA aquaculture model.
At this juncture, I just want to acknowledge the contributors to this study. The study was funded by Kenya Climate Smart. Thank you for listening.
Thank you very much. Any short questions for Esther? I know we've run out of time, but maybe 1-2 short questions.
Yeah, sorry.
Any short questions? Okay, mine will just be maybe as an additional pointer, because I know in Kenya, mariculture has not really gained traction compared to aquaculture. So maybe when policymakers and scientists like yourself, myself and others, come up with a management plan for the mariculture sector, I think this is one of the things that could be recommended, because it seems to be feasible and could help also in the livelihoods of people, and because it's a high valued commodity, the sea cucumber, also help in the exports and revenue generation.
Yeah, sure. Thank you.
You're welcome. So I think that marks the end of our first session. We are supposed to have a short break before joining the main session at in the next 20 minutes. I believe we shall meet back again in the afternoon, and I hope by then we shall stick strictly to the time. But in case of any question, please feel free to reach out to me via chat box.
Thanks, Stephanie. And if anybody would like to join the Women in Science tea break, we've put the link in the chat. So head over there to find out more about one of the other networks in the region. But otherwise, we'll see you later.
Thank you, Shannon.
Please, everyone, pick up the links that have been put on the chat because I'm going to end the meeting now.